This video will focus on the basic mechanism behind DNA replication and will also briefly introduce how mistakes can be corrected that might occur during the process of DNA replication. What we're going to focus on in this unit is the essential question: how is DNA replicated during the S phase of the cell cycle? Recall that this refers to the synthesis, or the DNA synthesis phase, that has to occur before cell division can take place. In other words, chromosomes in your cells have to replicate themselves before cell division takes place. And so that's what we're going to focus on here in this unit. How does this process of DNA replication actually occur? There are essentially three hypotheses that were explored early on regarding the overall mechanism of DNA replication, namely the semi-conservative, conservative, and dispersive replication models, each of which is illustrated in this lower right-hand panel here. There's an elegant, intriguing, classic experiment that addressed which of these three modes of replication actually occurs in living cells. And I'm not going to take time at this point to actually go through the details of this experiment, but essentially it's referred to as the Mendelssohn-Stahl experiment, and it's illustrated in this panel here. I do encourage you to work through and understand how these researchers were able to make explicit predictions for each of these three models of DNA replication, and how the results of their experiment show pretty conclusively that indeed DNA replication in living cells occurs by the semi-conservative model illustrated toward the left-hand portion of this image. So in essence, what this refers to is that if you consider a piece of DNA in a living cell, the two strands of that piece of DNA separate. So the hydrogen bonds are broken that occur between adjacent bases in the DNA molecule. Two strands separate, each strand of which is used as a template, so that complementary bases can be added, resulting in the formation of two genetically identical pieces of DNA, genetically identical to each other as well as to the piece that we start with. So again, this illustrates the Mendelssohn-Stahl experiment. Again, I won't go through the details at this point, but again, bottom line is DNA replication occurs in a general sense by this semi-conservative model. Let's explore this in a bit more detail. One of the things that I want to point out at this time is that imagine the two strands of the DNA molecule separating, and then complementary bases are added to each of those strands, but it turns out that the new DNA bases are added only in a 5' to 3' direction. Recall that DNA we describe as being anti-parallel, meaning the two strands of DNA run parallel to each other but in opposite directions. And also recall that the 5' and the 3', that just refers to carbon numbers, so carbon number 1, 2, 3 in the sugar portion, the deoxyribose portion of DNA, carbon number 4, carbon number 5. So the 5 and the 3 again just refer to these carbon numbers, and this refers to the overall directionality of each strand of DNA. So again, the basic idea is that as new bases are added to an existing template strand of DNA, they're only added in a 5' to 3' direction. So what are the implications of this? What I've done here is sketched out a very small portion of DNA that's unwinding, or I should say separating, in a right-hand to left-hand direction. So toward the right, the two strands have been separated, and this will continue as we move toward the left of the image here. So we're exposing two single strands of DNA, exposing the bases on each of those strands. And again, the new bases that are added are only added in a 5' to 3' direction. So keep that in mind. One of the things that has to occur first is that a primer has to be produced. In other words, bases aren't just automatically added. A small primer has to be produced, and it turns out, oddly enough, that there's an enzyme that produces a primer, and it turns out that the primer, shown here in yellow, is basically a short sequence of RNA bases. So here we have a few RNA bases that are added. Now that provides a 3' carbon to which new DNA bases can be added, again in a 5' to 3' direction. So once the primer is produced, It attaches to a portion of an exposed DNA strand. Then DNA molecule or DNA bases can be added in a 5' to 3' direction, as we see here toward the top of this image. Again, the same thing is occurring in the lower portion of this DNA molecule, but think about this process. If new bases are only added in a 5' direction, just like we said before, we have to produce a primer to get the whole process started. So here's a piece of RNA that's been produced, attached to this portion of the DNA molecule. New DNA bases added 5' to 3'. But again, imagine as this original DNA molecule continues to open in a right-hand to left-hand fashion, at some later point we have to produce another RNA primer that will again attach itself to the DNA. New DNA bases will be added 5' to 3'. So think about this. Toward the top, we can add DNA bases in a continuous fashion. On this lower portion, we essentially add DNA bases and we get these sections or these chunks of DNA that are produced. So this poses some issues, potential problems that we have to address. First of all, at some point we have to remove these pieces of RNA shown in yellow. After all, when we synthesize new DNA, we don't want to have these RNA bases found intermingled amongst our DNA. Not only that, but we'll at some point have to attach, on this lower strand here, attach these sections of DNA that are produced. Because on this lower strand, DNA is synthesized in a discontinuous fashion. So these sections of DNA that we've identified on this lower strand, we call those Okazaki fragments, after the person who figured this all out. We can also refer to the leading strand and the lagging strand. So DNA replication on the leading strand is continuous. On the lagging strand, it is discontinuous, again forming these Okazaki fragments. So in essence, that's what's happening. But now let's explore this in just a bit more detail. So what we'll do here is explore some of the major enzymes and proteins involved in this whole process. We won't necessarily mention all of the proteins involved, but we'll just focus on some of the main ones, beginning with helicase. So imagine a piece of DNA, double-stranded. One of the first things that has to occur is that the hydrogen bonds between adjacent bases have to be broken, so the two strands of DNA have to be separated. So helicase does just that. Illustrated in light blue in this image, helicase is the enzyme that splits apart the two strands of DNA. Once those two strands are split apart, they would have a tendency to snap back together through hydrogen bonding between adjacent bases, but that tends not to happen due to the presence of these single-stranded binding proteins, or these SSBPs, shown in dark blue. So the SSBPs, they essentially prevent the two strands of DNA from snapping back together. As we said before, we have to have a primer produced to get the whole process of DNA synthesis to occur. So the primer, again, is just a small section of RNA, shown in yellow here, and the enzyme primase is the enzyme that produces that RNA primer. So at this point, we're all set and ready to go, ready to add new complementary DNA bases. So kind of a major enzyme involved in this whole process is a polymerase, a DNA polymerase, and specifically DNA polymerase 3. DNA polymerase 3 is one of several DNA polymerases that have been identified. DNA polymerase 3, shown here in green, this is the enzyme that once an RNA primer is produced, the DNA polymerase 3 can then come along and add complementary bases to existing bases on each of the strands of DNA. In other words, the leading as well as the lagging strand. So DNA polymerase 3 makes its way down along each strand of DNA molecule, adding new complementary bases, essentially forming two new pieces or two new strands of DNA. Also recall that we have some issues to deal with. What about those Okazaki fragments? And what about the RNA primers that we want to somehow remove? So here we have another enzyme. We'll introduce DNA polymerase 1, shown in yellow. DNA polymerase 1 is the enzyme that removes the RNA primer and replaces those RNA bases with DNA bases. So our DNA polymerase 1 adds DNA bases after it removes the RNA primer, but we're still left with this gap between these Okazaki fragments. So DNA ligase is the enzyme shown in light blue toward the bottom of this image. DNA ligase is the enzyme that essentially forms covalent bonds between these adjacent bases and thereby sealing the gaps between Okazaki fragments. So hopefully this whole process occurs without too many mistakes or too many errors. Again, this occurs quite rapidly during the S phase of the cell cycle, but certainly mistakes can occur. And so what happens when mistakes occur? Well, it turns out that DNA polymerase III has some ability to proofread. In other words, you can imagine this enzyme making its way down along the DNA in your cell. So imagine your chromosomes. We're talking billions of base pairs. For the most part, it's pretty efficient, always adding A's and T's and G's and C's, but again, mistakes can occur. So for example, if it accidentally adds an incorrect base, such as we see here in this top panel, what it will do is pause for a split second, remove that base, and hopefully add the correct one in its place. So again, DNA polymerase III can proofread and correct its mistakes, but there are also other changes to DNA that occur. For example, imagine all the various environmental toxins and so forth that organisms are exposed to, and as one example, consider ultraviolet light. We have all heard that too much exposure to sunlight can lead to skin cancer and so forth. So in essence, what happens? Well, it turns out that ultraviolet light can interact with DNA, and often when there happens to be two adjacent thymine bases, the UV light can actually cause covalent bonds to be formed between adjacent thymines. So we form the so-called thymine dimer. Well, it turns out that that changes the overall shape of DNA in that particular cell. That can cause potential problems and has an implication in the development of skin cancer. So hopefully, when these types of things occur, a whole series of enzymes involved in protection of the DNA are activated, and essentially those modified bases, those thymine dimers, are recognized and they're removed and replaced with normal, fully functional thymine bases. So again, in addition to DNA polymerase III correcting its mistakes for the most part and removal of damaged bases like we see here, there are some mechanisms in place to correct mistakes that occur either during the synthesis of DNA or even after DNA is actually fully synthesized and operating within living cells. Obviously, it's not fail-proof, and of course we have various diseases, including various types of cancer, that overcome some of these protective mechanisms. This concludes our brief discussion of DNA replication and repair.